Hi, thanks. We talked a little bit about enzyme replacement therapy. We're going to be talking a little bit more about it after this next talk. But ICV is a method of delivering enzyme replacement therapy for brain lysosomal storage disorders, and there is a drug that's approved that uses ICV for delivery of enzyme replacement therapy. So Jack Madden from UCSF is going to explain to us what ICV is.
So I'm happy to share our experience of enzyme replacement therapy via ICV in the real world. This will be a quick introduction and overview. I'll go over how ICV therapies have been used, why they're used, what an ICV device is, and how it works, and then how we approach accessing and using the device, and then talk about some risks and other considerations when using this approach to treatment. So this is our team. Dr. Chang, nurse practitioner Jazwiak, and myself. We are the ones who have been doing many of these therapies. Our experience is mainly with CLN2 and in clinical trials with a lot of the MPS disorders. So ICV therapies are used to treat central nervous system and neurodegenerative diseases. It's not new, it's not a new approach, but previously the primary targets were cancer or infections, and those are pretty time-limited conditions. But in 2017, the FDA approved Brineura for the treatment of CLN2, the lysosomal storage disorder. And then in 2024, I think the indication was expanded. But it's a long-term therapy. Gene therapies are in development, CNS-directed therapies injected directly into either the ventricles or the cisterna magna. It's a one-time administration, but we hope more of a long-term effect. And then there's enzyme replacement therapies in development for other lysosomal storage disorders, possibly GM1. Hope. So why is this approach needed? Because it's very difficult to get to the brain from the body. And we've talked a lot about the blood-brain barrier and the challenges of overcoming that junction. And it's the brain, the central nervous system is fundamental in all of our functions, and we want to protect that. So you want to keep things out that don't belong there. So that's a good thing, but when you want to get something in there, it's very challenging. So ICV administration going directly into the brain allows you to reach the target tissue. The enzyme replacement therapies that were developed and approved for the MPS disorders treat the body, but they don't cross over to the brain. There are studies currently ongoing that have modified the enzyme and the delivery vehicle so that it can cross into the CNS, but you need very high concentrations of drug in order to do that. And if the main condition that you want to treat is in the brain, and that you don't want to waste enzyme by delivering it into the body where it's going to be taken up into tissues cleared by the liver, and you're going to have to give a lot of drug in order to cross to the brain. So if you can get it directly into the ventricles, into the CNS, you're much better off. So it's relatively easy to give intravenous therapy. But if we're going to be looking at long-term CNS-directed therapies, we need to have a predictable, safe, and easy access for repeated dosing. So that brings us to the ports, or a device. So this is what we're using for our current MPS 3 trial. It's a small port. The top picture, it's about the size of a dime. And the depth is about 2 dimes stacked. So it's a very small device. And you can see, I don't know if the resolution is good on that screen, but this is one that we practice with. And you can see sort of needle poke holes in the middle of the device. And that's the access point. There's a very small, so it's half a dime that you're aiming at through the scalp and the hair to access this port. The underside, the bottom picture, shows a little divot, and then a little neck, a little handle that comes off the device that is not functional, but it does help to orient the device when you're trying to figure out where it is. Attached to that divot is this catheter. The tail end of that is fenestrated. There's little pores or openings. And so this is the total device. This is surgically placed. Children are under anesthesia. And a small area of the scalp is shaved. There's a semicircular incision that's made. There's a little pocket. A small hole is made through the skull. This catheter is threaded through into the ventricle so that that tail end with the holes is resting in the CSF, the cerebrospinal fluid. The access point is under the scalp, but over the skull. And the kids, we usually admit them overnight for observation after the procedure, but they recover very quickly. There's usually very little pain, and we're ready to use the device within a week or 2. So when we go to use it, we access the port, the part that's under the scalp but over the skull, with a needle. We'll aspirate a little CSF fluid. It's clear, it looks like water, it's not viscous. That confirms that we're in the device and it's functioning well. Then we'll switch out the syringe and inject whatever therapy, drug, gene product, whatever it is, through that device into the ventricles. I'm old school, that's paper. We go slowly when we do this. It's about 30 seconds to a minute per milliliter. If the drug is 5 milliliters of fluid, we will often take off 5 milliliters of CSF to keep it in balance. So this is our process. We would numb the skin over the port. 2 minutes, okay. We'll trim the hair. I jumped ahead. Okay, we'll trim the hair. We won't shave it or use hair remover because we don't want to irritate the skin. Everybody's sterile. Everybody's wearing hats and masks except the kid who gets a pass. We deep clean and allow that to dry for 2 minutes. We use a clock to time everything so we don't rush our procedures. And we use a special needle that minimizes the trauma to the port when we're accessing. So this is, the Smurf suit it's a sterile gown and sterile gloves, it's a mask, it's a hat for the person who's doing the accessing. This is one of our kids. He's leaning up against his father's chest. An assistant is helping to stabilize his head, but he generally does not flinch when we access. The numbing cream works. He's a very calm child. He's 3 years old. I'm very impressed with his ability to stay still. He watches his tablet, and that's good because he holds it, so his hands are occupied, so he's less likely to reach up and try and figure out what's going on. He can drink, he laughs, he falls asleep. It seems to be very non-traumatic. And then when we de-access, there's very little blood, just a few drops. It's much less than a blood draw or an IV. And this is a child who's having Brineura. So our procedures that we're doing now, it's a pretty quick, it's maybe 20 minutes. This is a Brineura infusion, which is much longer. So we do a different procedure. We'll wrap the head and leave the needle in place. I think the infusions now are about 4 hours. So it's a long time to be stuck in one place. So this gives the kids a little bit more maneuverability. So, okay. Risk and considerations, there are some, there are many, but I will say that it works to get treatment into the brain. It's not perfect, but it is one step on the way toward an effective therapy and perhaps on the way to a cure.